Hi, today I'm going to be walking you through DNA and how it's packaged, provided by Kansas Corn STEM. My name is Jessica Sadler, and I'm a 6th grade science teacher and lead teacher for Kansas Corn STEM. Today we're going to be walking through the lab, DNA, and how it's packaged. Let's get started. First of all, you're going to need some fairly simple materials. A Sharpie, some Scotch tape, 32 ping pong balls, that's going to be 8 per group, some Velcro, it doesn't matter whether you use the strips or the little sticky dots, and 2 55-gallon trash bags. You're going to start by taking the separated ping pong balls and you're going to add the Velcro pieces together. I personally like to do this before giving it to my students, but you could also have them do that as well. The ping pong balls are going to represent your histones. Once they are connected, you have created your first nucleosome. It should look something like this. When every group has created a nucleosome, you should end up with 4. At this point, it's time to get out the saran wrap. For your saran wrap, you're going to want about 10 feet. I like to use a little extra just in case I wrap the beginning and the ends a little tighter. So you're going to pull it off the roll, and then you want to kind of squish it and roll it where it's in a rope style form like this. Choose your first group's nucleosome, and you're going to wrap it with the saran wrap. After you've got it wrapped, it's a good idea to take a few pieces of that Scotch tape and attach it so that it doesn't fall off. By the time you're done, it should look something like this. You can then take the nucleosome for your next group. You're going to want to leave just a little bit of space in between. You don't want them right next to each other, and you're going to repeat the same process. Congratulations, you have now completed your first strand of deoxyribonucleic acid, also known as DNA. At this point in your modeling, you'll want to start taking this and making it in a more condensed fashion, pushing it a little closer together. Okay. You are now looking at your first chromosome in a more condensed fashion. From this point, you're going to want to grab that first 55-gallon trash bag. And you're going to add your chromosome inside of it. I like to fill it up with about half air. And then once it's at this point, you can either choose to tape or tie it off. I personally tie it off. That way I can ideally use the bag again for other hours. If you tape it off, sometimes the bags can tear. It's totally up to you. Okay. We are now modeling our nuclear membrane. From this point, we're going to take our nuclear membrane, we're going to add it into another 55-gallon trash bag. do roughly the same amount of air. This is your last layer, so if it's a little more full, it doesn't matter as much. And then again, you may choose to tape or tie off the ends. Congratulations, you have reached the final step. You now have a cell membrane. If you'd like to know the effectiveness or the cost that it might take to do this lab, it's broken down for you right here. The Velcro is going to be about $10 depending on the kind that you pick. You can also do about $3.50 for the tape. Ping pong balls, Brewski Brothers, you can get 144, and it's about $6.45. The Saran Wraps, roughly $3.50. The 55-gallon trash bags, if you get the HDX version, they sell them at Home Depot and plenty of other places, Lowe's as well. Those are going to be about $10 for 50 bags, so you'll have plenty to do with other hours. And then, of course, the Sharpie is going to be about $1 if you don't already have one. For part 2 of this lesson, now that we have a model, you're going to want to talk, maybe have a Socratic seminar or just some discussion questions before about DNA in itself. For instance, you might ask your students, what are the barriers from keeping us being able to get to the DNA? Ideally, they should be able to come up with the cell membrane and the nuclear membrane. You might also ask them, okay, what are the cell and nuclear membranes made out of? Ideally, by doing research, or perhaps they already know, those would be bilipids, also known as fats. You might then move on to them doing some research about how do you begin to possibly break those things down, and then how do you break down the histone proteins that are still attached to the DNA at the end. Once you've done that, you might even extend and have your students talk about, is DNA the same within plants as it is within animals? Again, these can be done in advance, or you might choose to stall and do them towards the end when your students have more background knowledge. Moving on with part 2, you're going to be doing a CER writing with your students, also known as claim, evidence, and reasoning. I would start by asking them, how can you get the DNA out of the cell and then out of the nucleus? For my students, I like to start and give them a sentence stem for their claim because we want to promote the idea that it's not just a yes or no answer. So we have started with, the DNA can be removed from the cell by, and then they would put in the rest of their claim, and then they would continue on with the second part. It can then be removed from the nucleus by. Once they have set up this part, it's a really great idea for them to begin collecting their evidence and ideas for their reasoning. So we're going to get started. Separation techniques, as you can see, we talked about the bilipid membranes. How do you open that cell membrane? How do you open that nuclear membrane? Because they are a fat, the easiest way to do it is with soap. You'll see here in front of me, I have melted some butter. I usually use about 2 tablespoons, which is more than enough. You can shrink it or make it better, larger, whatever your needs are in your classroom. You can also do this if you get a vegetable or a corn oil and have that mixed, and then you can add a little food coloring to it so that you can see it a little bit better. Dawn dish soap, and you will be able to see the same thing. For today, we've got the butter, as well as some regular Dawn detergent dish soap. And I'm gonna come in really closely, and I might just make a little stream of it right there. And you can see as soon as that detergent touches those fats, they instantly start to part off to the sides. You can make it larger. Sometimes students think that it's happening just from some of the weights, but when you get it in there, you can see that the detergent is actually causing the separation of those bilipids or those fats. So that's a quick and easy way for them to model and see that up front. I like to have them take pictures or maybe even a video of the process, slow motions really nicely, and then they can be able to use that same information when they're doing the evidence part of their claim, evidence, and reasoning. The next thing that's super helpful is once we've gotten through those membranes, how do we get those histone proteins to release? And we're going to do that by salting them out. To salt them out, you're going to want some whole milk, salt plus some water, a salt solution. I personally like to make sure that when you have the salt solution made, it's got a good amount of salt in it, but it still looks somewhat clear. If they can see it too well, sometimes they think that it's just the salt falling to the bottom. They get confused on what they're actually looking at. So it's nice if you've already got it mixed up where it's fairly clear. You're gonna wanna add 50 milliliters of the salt solution to warm water so you get a nice good mix on it. And then you're gonna wanna add about 50 milliliters of whole milk. I've already pre-measured these out into cups so that they're ready to go. Okay, we're gonna add them together. And then we're going to stir the mixture for about a minute and then let it set for about 5. Now that we've let this set for 5 minutes, when you hold it up, it's a little easier if you get a good light source behind it. And I'm going to hold it, I don't know if you can quite see, but when you get it held up to the light, you can start to see a separation difference right on the top of your solution of where that is starting to move and become, those histone proteins are starting to be separated away from the rest of the mixture. This is also a great time to have them record the evidence either through a photo, a video, perhaps a slow motion video again might be kind of nice. Definitely we're going to be close up with this so that they can really see that layer or that barrier that's there. And then that would be another excellent piece of evidence to use. Once you have some of those evidence pieces, you would want to go back and finish writing that claim. Have the students provide the evidence in a very factual way. When we did this, these are our results. When this happened, these are our results. And be done. The reasoning is where you add that thought and that extra background knowledge to your evidence. So this is almost, sometimes I even let my students put it in a slightly more bulleted point fashion so that when they get to the reasoning, especially in middle school, they don't always understand the difference between these two. And so we talk about how this is where you add the extra bones and pieces to your argument and you kind of clean it all up and make it a very sound understanding. When this is complete, I have students turn it in and then we move on to part 3. Let's get started. Okay. For part 3, we're actually going to be extracting DNA from canned corn. Again, you're going to need some simple materials, one being a can of canned corn. You're also going to want a beaker. At least 200 mils is probably preferred. These are 500 mils. These are what I have in my classroom and they work really nicely. I like them. You're going to need some salt and you're going to want a blender to start this process. Okay. I've already pre-measured out 100 milliliters of canned corn. I do strain it a little bit of that excess juice off so that there's not too much in it. We don't want it too soupy, but you don't want it extremely dry either. We're gonna dump that into our blender. You're also gonna wanna add 200 milliliters of water. And somewhere in the ballpark about 1/8 of salt. This mixture is going to get blended for about 15 to 20 seconds. If it still looks pretty thick, you might want to go a little bit longer. And once it's done, you're going to be putting a strainer over a plastic cup and you're going to pour your corn, that soup mixture, into the top of the strainer. You'll probably have a little leftover. What I have students do is fill it, wait a few moments, and as it starts to settle, they're able to add a little bit more. You'll want to let it sit until there's not a lot of soup or drip coming out. So this has been sitting for a little while. You can see that most of the moisture out of my mixture has come out, and I just have this piece left. During that 5 minutes, it's a great idea to review with students. That might be a time where you have them go back and work on pieces from their claim, evidence, reasoning if they didn't finish, just so there's not 5 minutes of idle time. After the 5 minutes, and there's nothing left coming out, you're gonna wanna set this off to the side. And you're gonna be adding about 2 tablespoons of liquid detergent. Again, Dawn dish soap. You're going to give this mixture some gentle swirls. You don't want to do it too aggressively, otherwise you'll start to create bubbles and we don't want that. Once that mixture has been swirled well, you're going to let it also set for anywhere between a couple minutes just to kind of get it ready to go. And then you're going to want to put some of that solution into a test tube. Within this tube, you're also going to add a pinch of meat tenderizer. I like to put the tenderizer in first and then add the solution so the tenderizer doesn't get stuck to the sides of the tube. This one has been done. Once it's set like this, you are ready to add your ice cold, must be ice cold alcohol. Okay. You can either put the bottle in the freezer for a hunk of time. It's not going to do anything, so I put this whole bottle in the freezer usually on the day that we do this lab. You can also pour it into a different container and set it on ice, whatever works best for you. When you go to add the alcohol into the test tube, you might use a disposable pipette, like what I have here. You're going to take your alcohol and you want to hold the test tube at an angle like I am here. When you add the ice cold alcohol, you want to make sure that you're adding it down the side of the test tube nice and slowly. And the goal is to get about an inch that is sitting on top of your solution. So sometimes I do a bit and I'll just double check and see how that's panning out. So right here, you might be able to tell I don't have nearly enough sitting on the top of this. So I've had a quick check. I'm going to go back and slowly continue adding more. Perfect. Okay. Once you have about an inch of the alcohol sitting on top of your solution, you can go ahead and cap it off. And then you're going to want to let this sit for anywhere between 5 to 10 minutes. I like to go a little bit towards the longer side just because it allows for a little bit more separation to take place. The solution's been sitting for a pretty good amount of time. It may be hard to see on the camera, but if you I'll hold it on an angle. If you look right here at the top layer of your test tube, you can start to see that DNA has risen and is sitting on top of this layer right here. It's going to be a fairly light colored layer, and it will rest right there. You also want to encourage your students to be really gentle when they're picking up and moving this, otherwise they're going to break up that layer and then they're not going to get very good results, if any. At this point, you may also take a pipette if you'd like to extract some of that DNA for later or to keep. Gently down to that first layer. And you can extract a sample of it up. You can also put it in a separate test tube for later if you'd like to keep it for other reasons. Thank you so much for taking place in DNA and how it's packaged.